Hey everybody, this is Miss Segovia and I'm going to walk you through your notes on translation. For this activity you're going to need our handouts. One is the ribosome handout right here that looks like this. From our last activity you're going to need your strand of mRNA, and finally you're going to need to have cut out all the amino acids and tRNAs that are going to be used. So let's start in your notebook with doing the notes, and then we'll come back to the cutouts. So this process here is known as translation. In our last one, we talked about transcription. Right here, transcription. This one is translation. And the easy cheesy way to remember which one comes first is transcription has a C and translation has an L. And C comes before L in our alphabet. So let's go ahead and do our notes. Translation is the process that creates a protein from the code on the mRNA. So we created that mRNA molecule in our last activity of transcription, and now we're going to read that code, or we're going to read it in the language of amino acids. So let's go through our steps. In the first step, this one happens in the cytoplasm. The small subunit of a ribosome attaches to the mRNA. Now what I'm teaching here is a little bit more advanced version because I know you guys are pre-AP, so I want to make sure you have the advanced knowledge. And so to draw that one, I'm going to draw my little strand of mRNA. Remember, it's single-stranded, so we'll show those bases being exposed. And our small ribosomal subunit just kind of looks, we'll just draw a little disc right here to show that it attaches onto the bottom. In the second step, the tRNA carrying the amino acid methionine, that's that one, methionine, attaches to the mRNA at the codon AUG. So remember, codons are those three-letter codes, and so the AUG, adenine, uracil, guanine, is always going to be the start for every single protein that we create. So let's go ahead and sketch that in. So here's my mRNA, and there's my exposed nucleotides showing our single strand. Got my small ribosomal subunit right here, and my first tRNA that locks on right there, carrying an amino acid, I'll put a little M in there, locks on to the code AUG. Now over on your handout, let's take a look at what's going on over here. So we're showing our ribosome, and our process is translation. In case you can't read it, translation. And down here we've got a little model of a tRNA. So on the top of tRNA, these little colorful bubbles, this is our amino acids. Each tRNA carries just one, and it's always going to be specific. This guy right here is our tRNA. And on the bottom is what we call an anticodon. And so this is the opposite code that matches up with the mRNA code that we already have. So if you want to, you can cut the little slits out here. I'm just going to let it go for right now so I can do the activity with you. But if we look at our mRNA code, we have AUG. So what you want to do is you want to find the tRNA with the complementary code on its anticodon. Oh, that's a lot of vocab. All right, so what's going to match with AUG? Well, let's see. Adenine is going to match with the uracil. Uracil is going to match with an adenine, and guanine is going to match with a cytosine. So if I have AUG on this side, on the opposite side, I should have UAC, because A goes with U goes with A, and G goes with C. So you would take this little tRNA right here, and you're going to glue him or tape him onto this spot. But then we need to identify the amino acid that goes with him. Now, I already told you it was methionine, but there's also another way we can determine that. If I look at my code AUG, we actually have a cool little codon chart. And the way we read this chart is we find our first letter here, which is A. That tells me my answer is somewhere in this column. My second letter is U, which tells me we're in this, so this row, this column. And so we're intersecting right here on this box. And then my last letter is G, the third base. And so if I come back over, it is methionine. And that's how we identify which amino acids we have. So our first one that locks in is always going to be methionine. And it's kind of funny because AUG is kind of like August, and so you can think about what month does school start, and that is August. All right, let's continue our notes, and then we'll come back to our little drawing there. Or not the drawing, but the handout. Oops, sorry. So here on step 3 then. So we've got our methionine locked into place. That little tRNA is holding it. And so the large ribosomal subunit attaches. It's kind of like we're assembling our little machine. So on this next one, when you sketch it, we've got our little mRNA with its exposed nucleotides. And our small subunit is still there. My tRNA carrying my methionine is locked in, but then the large ribosomal subunit comes in and locks onto place. And so this is what we call our initiation sequence. Sounds fancy, doesn't it? In step 4, the ribosome moves along the strand and new, get that all in there, and new tRNAs enter according to their code. And acquire the growing amino acid chain. We also have our empty tRNAs, because they're actually going to pass the amino acid on. I'll show you that in a second. Empty tRNAs are released to pick up more amino acids. They actually let go, and then they'll go back and they'll grab more, and they'll come back, and they're like little assembly workers. Like, all right, I dropped off my box. I'm going to go get another one. Cool, dude. I'll see ya. Come on back. So here's my mRNA again with my exposed nucleotides because it's single-stranded. Aren't you glad I'm not making you write all the AUGs? So my little ribosome, it started out over here. It's going to start sliding along. So if I draw my little ribosome, my large subunit, my small subunit, and I've got a little tRNA locked in here. He's going to be picking up all the amino acids as it moves along, and a new guy will come in and lock on carrying an amino acid, and as this guy moves this direction, he will pick up the rest of the chain. Now it's kind of like this poor little guy just got beheaded, and so when he's empty, he'll get kicked out. And he can go off and he can go get some more tRNA, or he is a tRNA. He can go pick up some more little amino acids because there's different amino acids out here, and then he can come back and be ready to lock on to the next position. And the whole thing just slides along like clockwork, and it builds this long strand of amino acids. And so if we go back to the handout that you have, the foldable that's going in yours, we can go ahead and use our codon chart to figure out what the next amino acid would be. And so down here, I've got guanine, guanine, uracil. And so if I got GGU, that means my tRNA has to be CCA. And that's going to lock on right there. Now, what amino acid is it carrying? We got to go to our codon chart. And so we always read the mRNA. So if I got GGU, if I look at my codon chart, I've got G over here, which means we're in this row, G up above, which means we're in this column, and U means we're this first one in that box. And so it's carrying glycine. And so I can go grab my little glycine amino acid and stick it on there. And then you would find your next one, GUA, and then UGG, and then CGG. We always, 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 always read the mRNA. Don't worry about the anticodons up here. You'll always, 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 always read the mRNA. Don't forget that. It's super important. All right, last step down on the bottom here. Step 5. So this has to end at some point. It just doesn't go on forever. And the ribosome doesn't slide off the end. It'll actually reach a stop code. So when the ribosome reaches a stop codon, and by the way, if you look at your chart, you'll notice up here UAA, UAG, and UGA all say stop. So when it hits any of these 3 codes, it's like, boop, you're done. You're over. Finished. So when it reaches that stop code, it is released, and the new protein is complete. It's kind of cool too, because the whole thing kind of just like falls apart. And so we've got our mRNA. Now sometimes another ribosome will lock onto the mRNA and it will keep reading it. Other times the mRNA gets recycled and we use those nucleotides to build other RNAs. The ribosome would have reached the end, so our small subunit and our large subunit let go. And our new protein, so all those amino acids, so that guy is my protein, it's done. And so let's go ahead and label these two. This is the small unit of the ribosome. This is the large unit of the ribosome. So this guy here, that guy there, that's my mRNA. All those little guys, they just kind of disassemble. But my new protein here, it can get picked up by the endoplasmic reticulum, put into a vesicle, and sent to the cell membrane to become something interesting. Might be a protein that we're going to use inside the cell. Lots of crazy stuff happens, but the important thing is that it was created using this process. So what I need you guys to do, now that you got your notes all done, if you haven't already, go ahead and finish this. You'll want to tape or glue down all these little guys. Unless you want to keep all the spare parts, you can put them in an envelope, but it'll get kind of messy. But get it all together, and then this little one here is the only one that definitely does not get glued down, because when you go back and you study, you want to go back and look at the other pictures and use this one to remind you of transcription versus translation. So that concludes that, and I hope it was helpful. Thanks.